welcome to ImmunoTea time. Today we have a special guest host, Eva, who normally does editing. Right?
Hello everyone, I'm Eva. Usually I do podcast post-production, but today I am a voice here.
And today we have our special guest. Have you ever wondered who cleans up all the messes and infections in your body and kickstarts repair? And when bacteria get into your body, how are they removed and killed? The answer is macrophages, and our guests today help to elaborate how these cells in our body do their job. Professor Christian Bogdan's research in particular focuses on Leishmaniasis, a neglected tropical disease which affects hundreds of thousands of people each year. Leishmania actually invades macrophages and lives inside them, avoiding the immune system, but we'll hear more about that later. Professor Bogdan has a medical degree, he's a research group leader, a full professor, and was a member of the German Standing Committee on Vaccination against diseases like hepatitis B and COVID-19 for over 10 years. Welcome to ImmunoTea time, Professor. So today we have some very special tea from Russia, all right? So could you explain the tea to us?
Yes, I usually used to harvest... I used to harvest this type of grass where I'm from, on the forest of Russia. It's called Ivan-chai, and in English it's fireweed. So we have a mix today of black tea, fireweed and thyme.
Oh, it smells good.
We found very nice equipment in your kitchen. Very convenient.
Yes, the tea today is brewed courtesy of the Microbiology department here at FAU. I wanted to start with your time in the army, where you served as a paramedic and a nurse. Is that correct?
That's correct. Yes.
And so was that part of your mandatory service in Germany?
Yes, indeed. At that time, there was a obligatory military service, which was 1.5 years. And I actually decided to stay in the army for 2 years in order to use the time for something that could in fact be relevant for my future occupation. But at that time, I really have to say I didn't even know what I'm going to do afterwards. So the fact that I was joining the German army as a paramedic and got a training there was more or less decisive for studying medicine afterwards. Because medicine was not something that you were educated or trained in at school at all. So that was the only subject where I had no experience with respect to potential, study fields. And without that experience, I probably would have never studied medicine at all.
Well, so the circumstances, and then you decided to pursue medicine after that?
Yeah, because I had the opportunity to do part of my training in a private or in a civil hospital, not in a military hospital. So I was very much exposed to the regular patients in surgery and internal medicine, and I spent there 4 months altogether. And that was one experience which essentially made me think about medicine. And I actually got quite some positive feedback at that time from nurses and from doctors who said, why don't you study medicine? And I was thinking about chemistry and some natural science at that time.
Oh. Yeah, okay, that's really sweet that they said that to you, and then that pushed you in the right direction. And then you studied medicine at FAU. Right?
Well, I started then at the Friedrich Alexander University, and for the final year, in fact, I decided to leave FAU because I wanted to get some really in-depth clinical training, and I spent my entire final practical year in London. And in London, I was officially a student of the Middlesex and University College and Middlesex Hospital Medical School, which no longer exists today because they have more or less amalgamated their 12 medical schools down to 3. And this was a great experience because the training in England was very much focused on clinical expertise and examination and also questioning and taking history of the patients rather than using a priori the latest technology equipment. So at that time, the key to medical diagnosis in England was, and still is, to take the history of the patient and to examine the patient. And that's what I really learned there in depth. And I also had a chance to then rotate to different hospitals because I also had to take, besides medicine and surgery, a third subject, which was pediatrics. And I also even had an opportunity to stay for 3 months at the Hammersmith Hospital during that period of time, where I was working in a department of rheumatology. So that was a not only great experience, but it was basically also the foundation for what I actually decided to do later, because I was very much exposed also to infectious diseases during these 12 months in England or in London, as it is, a melting pot of the Commonwealth. There were many patients coming from overseas and coming especially to London to the very well-known hospitals in order to get treatment. So I saw a number of very rare diseases and also quite a number of infectious diseases, because this was also at the beginning, or actually at the climax of the HIV pandemic, when I was there.
So then you decided to go into research from there?
Well, actually, after England, I had to take the exam still in Erlangen, so I was one of the few students coming back to take the exam in Erlangen without having worked in any of the Erlangen or nearby hospitals. And afterwards, I actually applied to the pediatric department here in Erlangen, and the senior physician there, who was in charge of the personnel questions, he actually advised me to do first research and then go to the clinical work. And that was then the reason why I started in the Microbiology Institute, exactly in this institute, and where I also had done my medical thesis beforehand, and was working as a medical assistant in training for specialty field of microbiology, infectious disease epidemiology. At the same time, I was part of a research group from Martin Röllinghoff and Werner Solbach, and was sort of combining clinical training and research work in the lab, and very much focusing on T-cell response at that time to Leishmania parasites, and also macrophage responses. Yeah.
So do you think there's a difference in researchers who are trained, firstly as medical doctors, versus people who come straight from the university background and then do a PhD and come into it from that?
I mean, I think the key difference is that if you are a medic, you're depending, of course, on the field of research. You might be, more familiar with the underlying diseases and the true problems in terms of diagnostics or treatment or pathogenesis. So you have an advantage in that you even might know the disease you are working on. And another point, of course, is that as a medic, you are always sort of trained in a bipartite fashion. Yeah? So, of course, there are some examples where people are sort of leaving clinical training and don't do any clinical work anymore. But I think the vast majority is still trying to combine both areas. And this has the advantage that you never know how the one side or the other side will develop. Yeah? So in case you come to a situation where you make the decision, research perhaps is not the ultimate goal, you can still move into clinic for 100% and vice versa. Yeah. So I think that's the major difference in terms of learning techniques and knowledge about basic science. This very much depends on your own initiative and your own commitment. So you might have studied biology, and of course you have let's say, better training in basic laboratory techniques, but you can also learn that as a medic, and vice versa. The natural scientists, they of course can also become very much familiar with clinical infectious diseases. Yeah? So we have a microbiologist here in the diagnostic department who is a really trained molecular biologist and who was studying biology in Erlangen, and he has now been working in the clinical diagnostic department for 25 years. Has set up, for example, at the late 1990s, the molecular biology diagnostic laboratory, and he's very, very knowledgeable, similar to a medic, because during all these years he has learned about all the medical aspects as well. So I always tend to argue that these are not two separate worlds, but they are open to everybody who is interested and tries to really commit himself or herself to the field and to the questions.
Right now you are a director of this institute.
Yes.
Can you tell us, like, was your career a straight path, or it was more ups and downs?
Well, I would argue it very much depends how you define a straight path. I mean, it certainly was a geographically rather curvy path because I did not stay here for all my life, but I was moving back and forth to Erlangen. And of course, I mean, there was a time, especially before when I started here, I dived into a completely new project. Because during my thesis, I was primarily working on macrophages and their anti-leishmanial defense mechanisms. And then I got this T-cell project. Of course, Leishmania is a classical parasitic disease where T-cells play a major role. In fact, it was the disease where many of the current T-cell immunological concepts have been first described. So let's say the Th1, Th2 concept, for example, the dominant role of interferon gamma, of tumor necrosis factor, of IL-12, of reactive oxygen species, of nitric oxide synthase. That was all described in the Leishmania model. So I was lucky to have a model which at that time was quite central to immunology research as a model. And when I started now with the T cells, I got this really challenging project to screen a recombinant Leishmania library, a phage library, with the help of T cells rather than with antibodies. So normally you use an antibody screening in order to test for immunogenic proteins. And we were convinced that T cells are the most decisive defense mechanism in leishmaniasis, and antibodies are not only secondary, but they can even enhance the disease. And therefore, we didn't want to screen for any proteins of Leishmania that are recognized by antibodies, but rather for proteins that are recognized by protection-mediating T cells. And that turned out to be a real challenge because at that time we didn't have these very nice modern systems of recombinant antigen expression yet, where you can easily purify the proteins. One had to really extract the proteins, and they were initially insoluble, and you had to dissolve them and to dialyze them in order to get rid of the toxic dissolvent. And that turned out to be a really big problem because we had the impression that part of the T cell response was blocked by the preparation of these antigens. At that time, I was also away from Erlangen for a total of 8 months, where I stayed twice at the Center for Molecular Biology in Heidelberg in order to learn how to express actually these proteins, how to handle the phages, and where I actually learned the basic methodology of molecular biology. And that was a kind of bumpy way, because there were phases where experiments, of course, didn't work at all. And my former mentor, he always said, you also have to have a bread and butter project in order to survive scientifically. And this was the case. So I still had a macrophage project. But in the end, that was certainly a time period where I was also considering whether this kind of career is exactly what I'm heading for. So in the end, I had some very positive experiences, and the most positive experience was that this kind of research environment was not only stimulatory, but it was personally very, very nice to work in, and it was a very friendly and open-minded environment. And this actually then also allowed me to and also motivated me to apply for a scholarship to the German Research Foundation, which at that time was a classical training scholarship for doing a postdoc, another postdoctoral period. It was 2.5 years, essentially in Erlangen, and then I went for 2.5 years to Cornell University Medical College in New York. And this was certainly then the most important time in order to make a decision about the future career. Yeah.
So in the US, you worked with Carl Nathan.
Yes, I worked with Carl Nathan directly because he in fact was looking for a postdoc, but I didn't know about this because that was the time, prior to internet and all that. So you still wrote conventional letters, and afterwards you could also write some faxes. Yeah? I even didn't know at that time when I was applying what is his current sort of research focus. Yeah? I knew what he has published, and that was accessible to me. And so I applied to the lab because I wanted to dive further into macrophage activation. And then he wrote back to me, which was really a letter which I am sometimes still thinking about because it was sort of illustrating to me. If you have not read everything in detail, you might even overlook a central field. And he wrote back here, he's no longer working on macrophage activation, but he's working on macrophage deactivation. And if that is of any interest to me, then I'm highly welcome to come and introduce myself to the lab. Yeah. And he had, in fact, published 2 papers on macrophage deactivation, but I did not see them. Yeah. And when I heard about this concept, I said, yeah, that sounds really interesting because in the 1980s, and 90s, the immunology was still very much focusing on the processes of activation rather than on the question how the immune response is shut down again. So yesterday we had this Nobel Prize announcement in medicine, and the 3 people who now got the Nobel Prize this year, they were focusing on the aspect of downregulation of immune responses, in that case with the help of regulatory T cells, which they jointly discovered. Although the concept is not new. I mean, there has been this concept on suppressor cells. And with the macrophages, this was a new concept because macrophages were kind of considered to be activating, to be active upon activation. So antimicrobial activity, tumoricidal activity, also wound repair, that was all related to activation. And there was not so much insight at all what is really shutting them down. And the lab of Carl Nathan, they had previously described transforming growth factor beta as one of the inhibitory or deactivating cytokines. Deactivating in the strict sense of the word would mean that it is still active if you add it in after activation, unlike to inhibitory cytokines which block the activation. So TGF-beta, and that was one of the projects then I was working on, was a cytokine that really could truly deactivate macrophages. Another one which at least partially can do that as well is interleukin 10. And the main job actually was, and then I was back in molecular biology, was the characterization and purification of a new factor, which was called macrophage deactivating factor produced by tumor cells. And that turned out to be another really difficult project because all the attempts to clone it did not work out. So we eluded a purified protein, from gels and got even N-terminal sequences, but it was not possible to really identify at that time this molecule. At that same time in the lab, there was another group working on the biochemical characterization of this newly discovered enzyme, nitric oxide synthase. It was not yet cloned. It was an enzymatic activity, and it was only partially purified. And Dennis Stür in the lab actually then purified the protein and characterized all the cofactors that are necessary. And another group in the lab was then cloning inducible nitric oxide synthase. And then my task was in fact to analyze the regulation of inducible nitric oxide synthase by different cytokines, and that's where we discovered the macrophage deactivating effect of IL-10 and further first characterized the effect of TGF-beta on inducible nitric oxide synthase, afterwards the question was, what am I going to do now? Because the time was over. I had a 2-year scholarship, and another half year was paid by the American lab, and then I had to make a decision. So I had offers to stay in the US, actually also outside New York, because that was kind of an issue. I had to stay all the time in Manhattan, downtown. In the end, I decided to go back to Germany in order to continue with my infectious disease microbiology training.
Maybe now this is a good point. We can jump into macrophages. So I guess we can go into what the main role of macrophages is, and the sort of the two phases they have in injury and infection, and what they do and how that's regulated. Maybe we could talk about.
I mean, the classical concept has always been that macrophages are decisive for, on the one hand side, antimicrobial defense. And at the same time, it was clear that you need some kind of macrophage activity also during early phases of life, like during the organogenesis. Macrophages were early on recognized to be involved in tissue remodeling and also tissue repair. And the major questions people were working on were perhaps threefold. I mean, one question was, what are the mechanisms underlying the killing activity? So what kind of mechanisms are utilized by macrophages to exert antimicrobial activity? A second point certainly was, what are macrophages really doing in the context of a interaction with T cells? At a time when dendritic cells were not yet discovered, macrophages were the classical antigen-presenting cells. So a number of people at that time were also working on antigen presentation and macrophage-mediated T cell stimulation. And a third group was in fact focusing on cytotoxic activity of macrophages. That were the three hot topics. And there was a time when people were also thinking that macrophages can be used as a tumor therapeutic strategy. And this is now actually kind of, there's a certain revival because we have learned now a lot about the different activation statuses. So when I started with macrophages, there was, as I said, the activation by interferon gamma, which was the central macrophage, or still is the central macrophage activating cytokine. And then tumor necrosis factor also came in as a co-stimulatory cytokine acting together. And we had the various inhibitory cytokines that are shutting down the activation. But later on, it became clear that there are many more statuses of activation. So people used all kind of nomenclature then. So we are talking about the time when, in the 90s, when the so-called alternative activation of macrophages was described. So classical activation was interferon gamma, TNF, and people have actually noticed in the 1990s that interleukin-4 can also activate macrophage. I have to say, during my first postdoc period, we made a very interesting observation, which was more or less not taken notice of because it was unexpected. We observed that interferon gamma and IL-4 can synergistically act on macrophages. And people didn't want to hear that because interferon gamma was the newly described classical Th1 cytokine. So first in 1986, described as a classical product of Th1 cells then, and IL-4 was the counterpart, so produced by Th2 cells, and it took quite a while to then get this accepted, and And Simon Gordon in the US, in England, then found that IL-4 can also cause a certain status of activation, not in the context of interferon gamma, but rather enhancing capabilities such as wound repair, also the ability of macrophages to upregulate, for example, scavenger receptors that were IL-4 dependent. With respect to the antimicrobial effector mechanisms, NADPH oxidase, that was already known and is a classical effector mechanism, mainly also shown by neutrophils and macrophage and monocytes as well. And then we had this, newly discovered L-arginine nitric oxide synthase pathway. And this then opened up a complete new field, and that was due to the fact that nitric oxide is not only an antimicrobial effector molecule, but also has numerous abilities to modify protein. So it can cause nitrosylation, and it can cause secondary nitration, because nitric oxide is oxidized to nitrite and nitrate. And these chemical modifications have a huge impact on signaling cascades. So many of the signaling processes are susceptible to regulation by NO due to nitrosylation processes. And tyrosine nitration is another possibility. So that became a completely new field. And with respect to the different types of macrophages, people over the years have now used different nomenclatures. So we had classically and alternatively activated macrophages. Then another group called it then M1 and M2 macrophages based on this Th1, Th2 concept. And now the consensus is a little more, let's say, sophisticated, because we are not talking about fixed subpopulations, but we are talking about activation statuses that are irreversible. So we have seen ourselves that you can, let's say, deactivate a macrophage, and this macrophage is still susceptible to subsequent activation. So it's not a fixed status, it's not a subpopulation in the strict sense of the word, it can be reverted. And that has become now very diverse, because the numbers of mediators, also microbial products, which have a huge impact on macrophages, has led to quite differential responses.
Can you maybe tell us current status of macrophage inhibitor for treatment of diseases? Is it used? Is it researched?
Well, it's a little complicated, because your question kind of implies whether we have macrophage-specific inhibitors, and this is tricky. I really have to say that, because you can interfere with the regeneration or the generation of macrophages if you, for example, block MCSF or GM-CSF mediated signaling pathways. But to let's say selectively block macrophages as a whole, so the entire macrophage function, I'm not aware of let's say a single mechanism. I mean, we have, as I mentioned before, the possibility to downregulate certain macrophage functions. But think about IL-4 or IL-10, you can use it for blocking certain effector functions, but at the same time you are also activating certain functions of the macrophage. So a complete blocker is not available, but if one would think now about diseases, is there a disease where I would sort of want to have a macrophage inhibitor? There's only one disease or one disease complex where this would be desirable, and this is the huge complex of macrophage activation syndromes. So we have primary lymphohistiocytosis, where macrophages become overactivated, And this overactivation is really detrimental because it can essentially lead or will lead, because it's genetically determined, it will lead in the end to the destruction of all the cell lines in the hematopoiesis. So you get pancytopenic, so leukocytes go down, thrombocytes go down, patients become anemic, and in such a situation you might wish to have a blocker of this hyperactivation situation. Yeah? But in all the other situations, for example, antimicrobial defense, tumor defense, you would rather want to have perhaps a stimulation of the macrophages. There's a situation which we call fibrosis. Part of fibrotic syndromes can be due to the overshooting macrophage responses, or to the conversion of macrophages into fibroblast-like cells. That would be perhaps a situation where inhibitors could be good, but I think people are mainly at the moment focusing on inhibitors of signaling cascades rather than taking out the function of an entire cell population. It would also have a lot of side effects, because if you block all the macrophage functions, then you might also have an issue with your antimicrobial capabilities. And infectious disease susceptibility would increase. So insofar, I'm not aware that people have, developed something that would selectively block macrophages in total. You can block phagocytosis, of course, durch agents that are attacking the microtubules. That has been used for a number of years, not only experimentally in the lab, but we have some compounds like colchicine that have even been used in some diseases like gout and things like that.
How about with the inducible nitric oxide synthase? That enzyme is the main antimicrobial effector of macrophages, is that right?
Well, yeah, one has to be careful with the argument, what is the main mechanism?
Okay. Yeah?
I mean, the way to test it is twofold. You can take an animal model, a mouse model, and check out what does a mouse look like in the absence of inducible nitric oxide synthase. And you will notice then that this mouse has a problem to handle infections with intracellular pathogens, such as mycobacteria, toxoplasma, Leishmania. But for others, it's already becoming more complicated. Bacteria that are both in and outside host cells, for example, Salmonella, the phenotype is not so clear-cut. So there's also a huge contribution of NADPH oxidase. And for other bacteria, you might even find only a minor impact of nitric oxide synthase. And then we also have this long-lasting discussion. What is true for the mouse? Is it also true for humans? And there have been numerous papers published. It was like there were advocates who were arguing that inducible nitric oxide synthase is a mouse phenomenon and has nothing to do with humans. This is not only a misconception, but it's also due to the negligence towards a number of publications. What people have been doing, and this illustrates the complexity, they were using human monocyte-derived macrophages and then applying the same kind of stimuli that have proven to be good inducers of INOS in the mouse system. And it turned out that this is not really working in the human system. So if you take interferon gamma LPS and put it onto human monocyte-derived macrophages, the amount of nitric oxide produced will be extremely low. And that's due to some genetic differences in the promoter-enhancer region of inducible nitric oxide synthase between mouse and human. But there are other stimuli that have been described, and if you take, for example, patients with suffering from tuberculosis, you can find high levels of INOS expressed in the tissue. So one has to be a little bit careful with respect mouse-human, but in terms of the question, what is the most important mechanism, I think it very much depends on the particular infectious disease and on the tissue. So there's a clear organotropic difference. For example, something that is important in the skin is not automatically important in the spleen. We saw that also in leishmaniasis. So for controlling Leishmania in the skin and in the draining lymph node, you need inducible nitric oxide synthase. But in the spleen, the impact is rather small. Mm-hmm. It's only a 3-fold increase between wild type and knockout mice in terms of parasite burden. For others, it's the other way around. So, for example, reactive oxygen species and generation by NADPH oxidase is very important in liver and spleen, and much less so in the skin. Think about antimicrobial peptides. They are absolutely highly relevant in the skin, and that's probably also the reason why epidermal cells are such potent producers of antimicrobial peptides. And from that point of view, you have a particular defense situation on the outer surface of the body. But in other areas, the, let's say, inducible mechanisms are more prominent than the a priori existing or constitutive mechanisms. Then we also have this acidification of phagocytic lysosomes. We have nutrient depletion, which is also an interesting mechanism of defense, that you take away certain amino acids or other essential nutrients from a pathogen. For example, iron extrusion or efflux, or manganese or zinc ions, which are important for certain enzymes of microbial pathogens. We also have this very interesting mechanism of DNA extrusion, netosis, which again is not a mechanism for all pathogens. I mean, these DNA, let's say, fibers, it looks like fibers, it's very thin DNA structures. They are decorated with antimicrobial molecules such as elastase or certain histones, which interestingly enough can also have antimicrobial function. So I think it depends on the context, the tissue, the species to some extent, and also, of course, on the stimulation conditions that might be triggered in a certain infectious disease.
And then could you just, the thing that I found most striking, we had a lecture from you, was this story about the L-arginine and the possible host-directed therapy. Could you...
The L-arginine and the
host-directed therapy? The possible
host-directed therapy,
So could you... Yeah. I think that's a striking example of how we can learn how a particular cell type works, and then see if we can manipulate just a tiny part of that, and then see what happens afterwards. So could you...
I mean, this was a classical example where a clinical phenotype that you can observe with the help of a genetically deficient mouse can also contribute to the development of new strategies to perhaps even cure a disease. So the L-arginine metabolism is in fact very complex, because we not only have the nitric oxide synthase, we also have another arginine utilizing enzyme, which is arginase 1 and also arginase 2. And the observation was in the beginning that Leishmania, as a pathogen that is sitting and living happily in a macrophage as long as it is not activated, that the control of Leishmania in the mouse system is really completely dependent in the skin and in the lymph node on inducible nitric oxide synthase. Now, if you think about the fact that there are mouse strains that are not able to develop this protective immune response and cannot handle Leishmania, such as bulb C mice. These are the white mice that are frequently used, and they are certainly exceptional in immunology compared to the many other mouse strains. But unlike black 6 mice, they will not be able to control the infection. And that was actually one of the central questions in the 1980s, or actually already starting in the late 70s. Why is bulb C mouse susceptible to Leishmania major infection as compared to black 6? And then the first observation was the bulb C mice seem to develop the wrong T cells. And that was long before Th1 and Th2 cells were discovered. If you took T cells from bulb C mice infected with Leishmania major and you were transferring them into a nude mouse, you could in fact mimic this phenotype, so they again became susceptible. And the question was, why is that? And then we had this discovery of Th1, Th2 cells, and people argued, yes, in the bulb C mice you have more Th2 cells, more IL-4. IL-4 is, not a classical macrophage activator, but rather inhibitory, can counteract certain interferon gamma responses, and so on and so forth. And that's the reason why. And then we and other groups noticed that if you inhibit nitric oxide synthase or even delete it, then the mice will not heal. This was completely compatible with this Th1 concept because interferon gamma induces nitric oxide synthase. If you take away the nitric oxide synthase, you obviously take away an interferon gamma induced pathway, and the knockout mouse proved that in this case, answering your previous question, INOS is essential. So, and the next step then was why doesn't this happen the same way in the bulb C mice? There's a lot of discussion still going on and a lot of research what is really going wrong in the bulb C mouse, but the major observation then was that interleukin 4 is a key driver of arginase. And arginase is eating up the arginine that otherwise is utilized as a substrate by inducible nitric oxide synthase. And in fact, we could then show in my own group, so Uli Schleicher was actually the one who was publishing that together with me in 2016. If you take a arginase knockout bulb C mouse, then this mouse will no longer develop progressive disease. And now, more recently, we made the observation that leaving alone this funny bulb C mouse situation, which is certainly not, as I mentioned beforehand, the regular situation. If you go from Leishmania major to Leishmania mexicana, another Leishmania species, which probably more closely mimics the situation in humans, then for all of a sudden it's no longer an issue of bulb C versus black 6, because Leishmania mexicana causes chronic skin swellings in both of these mouse strains. And in the Leishmania mexicana, now we could show that the deletion of arginase again causes self-healing disease, so the mice can then really cure. And at the same time, we noticed that in the wild type situation, the mice actually develop a huge and chronic depletion of arginine in the tissue, because they massively upregulate arginase, and this massive upregulation of arginase leads to a degradation of arginine, and the tissue content of arginine in the skin, in the plasma, and also in the lymphatic tissue is going down. And you could even, or we could even state that there's a strict correlation between the arginine availability and the course of infection. And that actually was then letting us to postulate perhaps we can revert the course of infection by supplying exogenous arginine. This is a kind of tricky situation because you have a wild type mouse that is co-expressing arginase and inducible nitric oxide synthase at the same time. And as I just said, the arginase, due to the biochemical differences, can sort of deprive the INOS of arginine. But now if you feed that mouse with arginine, you could also argue that perhaps you're feeding not only INOS but also arginase. And even more complex, the parasite itself is also expressing an arginase enzyme, which is essential actually for the parasite replication, because the arginase in the end is the precursor, or the product of the arginase, the ornithine, is the precursor for polyamine synthesis, and polyamines are necessary for DNA replication. So when we first thought about this, I also said, if we are unlucky, the arginine might also feed, first of all, the parasite, more food for the parasite arginase, and therefore more parasite replication, or might even feed the host cell arginase, and therefore promote perhaps processes which are not at least related to antimicrobial activity. But this was not the case. So in the end, what Pablo Rai, who was a postdoc until recently in the group, then observed is that a early treatment with arginine starting at 10 days after infection can prevent the non-healing cause of infection, cause healing. But even more important, if you treat mice that have already a fully developed clinical disease and you switch then from normal drinking water on water with arginine, you can also induce resolution of disease. And therefore, our current concept is that arginine might really be a interesting host-directed therapeutic approach, because we also analyzed human skin lesions from patients with either localized or diffuse cutaneous leishmaniasis after infection with Leishmania mexicana. And they at least also express iNOS and arginase at the same time. Of course, we also have now to be careful, and cannot propagate right away that arginine is going to be the new therapy. So the plan is actually to use it as an adjuvant treatment along with classical anti-leishmanial treatments, which have a lot of problems. They are toxic, they are expensive partially, they are not readily available. They also have a long-lasting history of side effects. Insofar, yes, we cannot right away, advocate it as a new therapeutic compound, but we can use it as an adjuvant in order to see whether arginine treatment, and it's an amino acid which is freely available in drugstores, whether this can be used as an adjuvant to enhance or to accelerate the healing. And this requires, of course, a clinical trial now. The first goal now is to get this mouse study published, and once that is the case, I think we have sufficient data to move forward to the next step in order to see whether that could be a trick or a approach to improve the treatment.
Can you maybe tell us a little bit more about Leishmania intracellular parasite and which cells does it affect?
Yeah, it's actually very heterogeneous. The host cells are very diverse, and one really has to go back to the infection cycle. So Leishmania is transmitted by sandflies. So sandflies are little mosquitoes that are more or less expanding, similar to other vectors. You might have heard about Aedes, so the dengue and yellow fever transmitting mosquitoes, and sandflies are highly prevalent. Also in Germany, in fact, you can find sandflies in the entire Rhine Valley up to the lower Rhine area, so Cologne and even further on. And in these sandflies, the parasites develop in the midgut into infectious so-called flagellated promastigote forms. So these are the mobile forms. They have a flagellum. So Leishmania are flagellated protozone parasites. And the transmission is due to the fact that the sandflies that are harboring the parasites, they actually have a partially blocked gut. Essentially, there's an area where these promastigotes generate a gel-like substance, and this gel-like substance prevents the normal flux of blood into the stomach, in quotation mark, of the sandfly. So if a sandfly is infected, it has a hard time to successfully suck blood, and it becomes more and more aggressive because it's becoming more and more hungry, literally. And the consequence is that the promastigotes, which are sort of blocking the normal blood flow, are expelled. Yeah? So you essentially, if the sandfly bites you, the sandflies will transmit then also the promastigotes. Yeah? It's like a vomiting effect. Yeah? So they vomit into the newly generated wound, and the wound is also generated in a very specific manner, because they lacerate the small vessels at the border between the epidermis and the dermis, and thereby they generate a blood sea. And now the question is, if you sort of transfer Leishmania now into that blood sea, what is going to be the first host cell? And the first host cell, this has been shown also experimentally with the help of sandfly mouse infection models, but also in patients who have been voluntarily infected. The first host cells are neutrophils. And that's not so surprising, because many decades ago it has already been shown that neutrophils can successfully phagocytose Leishmania promastigotes, and they are numerically the most prevalent phagocytic cell in the peripheral blood. The next step is then again an interesting example how evolution actually acts and obviously developed in a way that the parasite has a survival or chance of survival, because the infected neutrophils, they will become apoptotic sooner or later, and the apoptotic neutrophils then can still harbor a viable parasite, because the parasite can shut down the antimicrobial, or possibly shut down the antimicrobial activity of the neutrophil, and therefore you have now a dead neutrophil with a viable parasite. And the dead neutrophil, as also shown for other dead cells, if you add dead cells onto macrophages, the macrophages will also become deactivated. Yeah, was actually also shown here 28 years ago by a former member here in the medical clinic 3. And this deactivation is an elegant way how the parasite now gets into a macrophage and is not killed, because the neutrophil shuts down the antimicrobial effector mechanism of the macrophage, and the parasite can sneak into the macrophage without being directly attacked. So essentially that's probably the key reason why initially after the infection there's not directly a elimination of the parasite, but the parasite doesn't really cause a lot of alarm, at least not to the same extent as let's say gram-positive or gram-negative bacteria, and has a certain chance to establish itself. And only if the macrophages then become activated with the help of T-cell mediated or T-cell generated cytokines, you might then develop a turning point. Neutrophils first sort of target cell, macrophage second. At the same time, we are talking about the skin. We are talking about the border between epidermis and dermis. So Langerhans cells have been described to carry Leishmania parasites. We also have dendritic cells as host cells, especially also then in later phases of infection, but also early on, even at day 4, you can already find infected dendritic cells, monocytes. Not only the tissue macrophages, but also monocytes are classical host cells. And after all, talking about the long-lasting persistence, like mycobacteria and Toxoplasma, Leishmania, even after treatment, they will persist lifelong. And one target cell that we have identified many years ago as a niche where the parasite survives are fibroblasts in the draining lymph node. There are many other mesenchymal cells that have anecdotally been described, not necessarily in the strict context of cutaneous or visceral leishmaniasis, but other mesenchymal cells, peritoneal cells, endothelial cells. There are reports, but I would argue that the key cells for understanding the pathogenesis are certainly the ones that I mentioned. Eosinophils, to finish with these granulocytes, are special. They can also phagocytose Leishmania, but there's currently no strict evidence that they are essential for control. they seem to be rather involved in non-healing or in the induction of non-healing disease. Neutrophils, by the way, as well, they have this antimicrobial activity, which is partially shut down. And in fact, if you take away neutrophils, it's more beneficial if you take the overarching evidence for the control of the parasite rather than for the survival of the parasite. Yeah.
So just maybe we can pivot a bit, talking about the actual environment of infection and how that results in what cells get infected. Maybe you could talk about the research group, the leader of the Immunomicrotope project. So what does Immunomicrotope mean?
I mean, the term immunomicrotope was coined, in fact, by myself in the context of a discussion we have had when Vahid Sandoktar and Georg Schett and myself were discussing topics for an excellence initiative. Yeah? So we were actually sitting together in an ICE train on the way back of a SFB evaluation. And I do remember that when Vahid Sandoktar was asking me, what do immunologists need to know in order to better, or need to measure in order to better understand what is happening during immune responses, not only during infections, but also, let's say, during autoimmune diseases or even during tumor diseases. Then I said to him, yes, we need to better understand what is really happening at the site of infection or at the site of inflammation or at the site of tumor defense. And I named a couple of examples, such as oxygen content, electrolytes, pH, tonicity, any of these organic or inorganic mediators. Because what do we measure normally? We take serum, we take plasma, we take blood. We might take bronchoalveolar lavage. We might take a swab from the skin surface. But other than that, we really don't know what are the exact microenvironmental and micromilieu conditions at the site of infection or at the site of inflammation in general. And then we were thinking, could this be, something for a new idea for an excellence initiative? And that was then the basis for the concept of immunophysics, which unfortunately failed. So we were not invited to write a full application, unfortunately, because the reviewers were quite happy with the concept, but at the same time said, we do not yet have enough preliminary data to demonstrate that the interaction between physicists and medics or immunologists might be really helpful, or that we can all measure all that, which I found a rather strange argument, because in an excellence initiative you should normally propose something that is directed towards the future, and not something where you already have lots and lots of data. Anyhow, and one part of this immunophysics concept was the immunomicrotope concept. And immunomicrotope, as the name says, it's an artificial word in a way, but I think it illustrates what the target is. So everything that is related to the immune response. Micro, everything that is related either to infections, microbiology, or micro in terms of imaging, microscopy. And topos, Greek, means the site. So the immunomicrotope concept is essentially proposing that the regulation and the outcome of an immune response, be it against a pathogen or against a tumor cell or in the context of an autoimmune disease, is very much dependent and influenced and regulated by the conditions at the site where the inflammatory response is initiated and goes on. And in order to rescue the concept, I then, after the failure of this excellence initiative, I then decided to use that concept for setting up a research training group. And in 2018-19, we then submitted that concept the first time to the DFG for a research training group, which is one of these classical funding modules of the German Research Foundation. And it was very positively evaluated in 2019 here prior to the COVID pandemic. But as I already mentioned in another context, it was not approved in the Senate. For unknown reasons. We did not get any reasoning. We got 10 pages of positive evaluation with not a single criticism. And then I started again, so handed in another pre and full application, and then we were successful. And right now, the idea is now to consolidate the concept in different infectious diseases and to define in bacterial, in parasitic, in fungal, in helminthic infections, what is really happening, at the site of the infectious process. And this is, of course, a challenge because we still have a lot of issues in terms of measuring, what is really directly the content of a compound X or Y in the interstitial fluid, for example. Yeah? I mean, we are not yet there. But, for example, what I mentioned before, mass spectrometry can be used to measure arginine in a skin lesion. So, and this is what we, for example, did. And there are several other examples now in this consortium where people have measured certain components of the microenvironment, of the micromilieu. Oxygen, for example, is probably one of the reasons why bulb C mice are not healing, because unlike two black 6 mice who develop a very nice vascularization in the skin lesion, it's very much reduced in the bulb C mouse, and therefore the oxygen supply is probably also negatively impacted. And Jonathan Jansch, who is a member of this consortium, who was a former PI first here in Erlangen and then in Regensburg and now in Cologne, he showed in his group in the Leishmania model that there is a true correlation between the availability of oxygen in the skin and the course of the infection. So if the lesion develops into the healing phase, then the oxygen supply is increasing in parallel to the nitric oxide synthase activity.
Yeah, I think it's great. I think for me, at least, the most interesting research is always multidisciplinary. Like it's at the intersection of two disparate disciplines, and you can bring them together and find something that no one else has looked
this
at.
Actually, to mention that briefly, is now the goal for the second phase of the funding, which we are applying for currently. And we have now this new center of Max Planck for Physics and Medicine, Max Planck Zentrum für Physik und Medizin. And it's certainly very good for the research training group that this center is now existing in Erlangen and is already working, and there are a number of groups that have started to work there. And Vasily Tsavodaev, who is the current spokesperson of the Max Planck Zentrum, he's also a member in our research training group. And one of the goals now for the second funding period is that we really intensify and extend the exchange with the physicists and the people at the Max Planck Center in order to make them not only aware of our questions, but also to find solutions for really analyzing the immunomicrotope in much more detail.
They had a symposium there a couple of weeks ago. I went to that. The stuff they're doing is really cool. Like the microscopy and everything. It's super cool.
Yeah, I mean, it's essentially the imaging is certainly one component, which is absolutely helpful. Another component, as I said, will be really the measurement of soluble components of the micromilieu. A third one is certainly the spatial transcriptomic strategy in combination with the proteome analysis, so that one gets an idea how does an infected cell look like in comparison to a non-infected cell, in terms of the transcriptome and the proteome. And of course, we also have to emphasize that many experiments still have to be done also in vitro in order to, first of all, delineate the impact of different factors on certain host cell types or on the parasite. We are also talking about certain metabolites, so products of the parasite, products of the host cell. For example, David Führinger's group is currently focusing on tryptophan metabolism because they made some interesting observation there in the helminth infection model, and this all requires, in the end, the combination of a multitude of different methods in order to better, define the complex composition of what we call immunomicrotope.
Now that we've finished our tea, we've got our last sip questions. So these are 3 questions we ask every guest just to finish the podcast. If you could go back to your first day in the lab and speak to yourself, what would you say? What advice would you give? Or anything?
You mean now retrospectively,
Yes.
based on my experience?
Yep.
Well, that's not so difficult. I would certainly say don't give up and think positively. And this exactly, was what I described at the beginning in terms of positive atmosphere. Yeah. So if I think everybody who has been working in science has made the experience that probably majority of experiments is not going to work. And if I say it's not working in immunology, it means the result is at least not, up to your expectations. And insofar, I would also emphasize one has to be careful with the statement, okay, the experiment failed. I would rather give the advice to say to have another look at the outcome of the experiment, because what sometimes appears to be a failure could in fact be an indication for an unexpected new result.
And actually, do you think there were times when you needed that like encouragement in the lab? There must have been, because we feel it sometimes just doing our classes, right?
No, I really have to say it's always a combination between encouragement and critical assessment. So there was certainly a time also in my scientific life where supervisors and mentors were kind of giving sparse sort of encouragement, because in the end, there was always something that was probably to be criticized. And I think for the new generation of scientists, it's really important that we still maintain our critical assessment. So, for example, you can be permanently positive and permanently encourage everybody and say, don't take it serious and it will be better next time. But I think it's also important to really figure out why, for example, did an experiment not work? What was perhaps experimentally going wrong, or what was not taken into account? Because otherwise we take it might be really dangerous to overlook obvious mistakes. And we have to develop a kind of sort of mentality that a critique is not automatically negative. I think this is something which I currently experience because the young students, they expect a lot of applause and a lot of encouragement, and might forget a little bit about the fact that we together, in a friendly and open and critical, self-critical manner, have to evaluate the outcome of experiments, or have to interpret the findings. And of course, if everything would be only negative, verbally as well as mentally, that's not good at all. But the other way around is the other extreme, which is also not helpful. So we have to have a balance between critical assessment and encouragement. And I think in the old days we probably had too much critical assessment, and currently my impression is we might have a little bit too much of encouragement, because if you're not criticized for something that you have done in a really wrong manner, and of course nowadays as well as in the so-called old days, we have perhaps co-workers who are kind of reluctant to advise. Yeah? That can be sometimes very good because they might make really interesting discoveries because they did it differently. But if 10 students and technicians have really demonstrated this is the protocol we have really to follow, and somebody doesn't want to follow that or for whatever reasons, you have to address that. So I guess it's the mixture, as always in life. Extremes are bad. Yeah.
Our second question would be, what is one mystery, scientific or personal, you hope to see solved in your lifetime?
Well, I mean, in a way, I'm also, a medic. I'm not only a scientist who only wants to solve basic scientific questions. But one really interesting question that we have been trying to work on for quite a while, and which is still not really solved, is the general, phenomenon of long-lasting microbial, or in our case, parasite persistence. And that's really intriguing because we can demonstrate that all the necessary effector mechanisms are available, and they even control this pathogen, because if we block it, then we get clinical reactivation. But at the same time, the parasite still manages to survive even after antimicrobial treatment. So this has two negative consequences. The one negative consequence is that you have an ongoing chronic inflammation, which could have ideally the positive consequence that you maintain memory, which is the case in terms of Leishmania infections, in the case of Leishmania infections, but it has also the negative consequence that you have a burden of pathogens that can elicit a clinical traumatic disease if you become immunosuppressed for other reasons. Yeah. So this kind of, why do we have control but no elimination, is one of the questions which I would love to get at least a further partial answer to in the remaining time of my professional life. And I mean, there are certainly also other questions, because it's not only Leishmania here in the institute. We are working in 10, 11 groups on different pathogens, and each of these pathogens has a probably similar interesting unsolved question. For example, in Q fever, which is caused by Coxiella burnetii, there is the observation that perhaps 1 to 2 to 5% of the people who become infected with this intracellular pathogen, which actually lives happily in a phagolysosomal compartment, in an acidic phagolysosomal compartment, which is really unusual. And 1 to 5% of people who become infected will develop what we call chronic Q fever, and we have no clue why. And that's really dreadful, because these patients require lifelong antibiotic treatment, and even then they will never ever be cured completely. So if you stop the antibiotic treatment, the danger is high that they will develop again overt clinical disease. So that's something which we don't understand. Why is that the case? And you can go on with others. For example, Plasmodium, which is another pathogen that is worked on here in the institute. There's this really interesting developmental cycle where the pathogen replicates in erythrocytes, and then there is a sideway where gametocytes develop. and why and how do these gametocytes develop in order to maintain then in the end the cycle via the Anopheles fly. Yeah? So there's obviously again probably microenvironmental dependent regulation, epigenetic regulation of genes, which then sort of cause this differentiation of plasmodium into gametocytes. But the environmental factors that are triggering that are absolutely ill-defined and not understood.
If you could ask the immune system one question and get a clear, honest answer, what would you ask? Like imagine the immune system is here.
Okay, what kind of question would I ask the immune system? That's a good point. I never thought about this. What would I ask the immune system? I might ask the immune system, why hasn't it developed in a way that it doesn't age? So you know this or heard probably about this phenomenon that we not only have the physical aging in terms of how we look like and disabilities in all kind of parts of the body, but we also have an aging which might be triggered by inflammation, but also simply by the time span of the life. And of course, it would be very beneficial if our immune system would remain, juvenile, because we are generating new cells, but there's unfortunately the event that the generation not only of new cells but also the activity of these cells is declining. So from my point of view, it would have been a good idea for the immune system to not participate in the overarching aging phenomenon, and I would ask the immune system, why did you decide to do so?
I want to know too. I wish I could. I should have an immortal immune system. Thanks for joining us. That was a good episode, I think.
Yeah, thank you very much. It was a very nice conversation.
Yeah, thank you for these quite challenging questions. And
you handle them very well.
Thank you.
Okay, cool. Reach us on Instagram at ImmuneFAU or send us questions, ideas, or feedback to immunefau@gmail.com. The equipment for this podcast is graciously provided by the Friedrich Alexander University Rundfunk Studio and Thomas Bauernschmidt. So I'm going to start with the introduction, and then we can have the tea, and then we can get into the interview proper.
So we have to have a cup of tea or what?
Yes. Yeah, it's the ImmunoTea Time podcast.
Okay.
So we drink tea and we discuss immunity. That's the whole
Okay.
fun,
I see.
fun part.